Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series, and the question we are going to discuss today is write in detail about the occurrence, transport, site of synthesis and bioassay of abscisic acid in plants, and also explain the effects of abscisic acid on various plants physiological processes. So this is the question, quite a bit of long question, but still we will do it. So here abscisic acid, the question is directly on abscisic acid, its physiological processes, occurrence, transport, site of synthesis and bioassay. And do not confuse here, ABA is the short form of abscisic acid. Abscisic acid is short is known as ABA only. Okay. If you don't know, although it is a common thing, most of you must know, but if you don't know, do not get confused here. Now let's move to the answer. So obviously introduction part, as the question is already asking 1, 2, 3, 4, 5 dimensions. I think if you see the questions, there are 5 dimensions in the question. So instead of writing a lot in introduction only, you can simply introduce that ABA is a naturally occurring PGR, that is plant growth regulator, and generally it is considered as inhibitor. If you see in the PGRs, there are promoters and inhibitors, and generally ABA and ethylene are known as inhibitors, whereas cytokinin, gibberellic acid, and auxins are known as promoters. So please do remember it. So I think if you write a 1-2 line of introduction enhancing on PGR, that would be more than enough. And it is your homework. On the basis of this framework, do write a good introduction and comment down, so that we can see how much you are brainstorming about the introduction part. So let's move to the answer now. So I hope you understand how to introduce. Now simply we will move to the body part, because there are a lot of things to be explained. So first of all, occurrence. So occurrence of ABA. So you should know that ABA is a naturally occurring plant growth regulator. Okay. So which we have discussed in introduction only, and it is classified as terpenoid. So okay, and it is highly distributed, or you can say uniformly distributed in higher plants. Okay, it is very much found in higher plants, and you will find these in leaves. You can find these in chloroplast of the leaves, especially in chloroplasts of the leaves. I'm mentioning here only. Let's save a bit of space, because we have to discuss a lot of dimensions. So occurrence, first of all, you can find them in leaves. You can find them in fruits, especially when fruits are developing seeds. While fruits are developing seeds, you will find the amount of ABA there. You can find them in root caps. We can find them in phloem tissues. Phloem tissues, we can find them in dormant buds and seeds. Why? Because ABA promotes dormancy. So if a bud is dormant or a seed is dormant, obviously ABA will be there. Dormant buds and dormant seeds. So these are the places where ABA can be naturally found. Secondly, we will now move to the site of synthesis. Second part is site of synthesis. So actually ABA is primarily synthesized in these regions only. You will find them being synthesized in green leaves, in roots, in developing seeds, etc. Okay, generally in green leaves, roots and developing seeds. So if you see its biosynthetic pathway, biosynthetic pathway. So it is synthesized from carotenoids via MEP pathway. It is synthesized through, or you can say from carotenoids via MEP pathway. What is this MEP pathway? It is known as methyl. It is known as methyl erythropetrol phosphate pathway. I am writing the spelling, difficult to pronunciate. Methyl erythritol pathway. Okay, or sorry, methyl erythritol phosphate pathway. Phosphate pathway. MEP. So remember this short. In short, if you do not remember the full form also, you just remember MEP pathway. That would be enough. So what happens? It is synthesized from carotenoids via MEP pathway, and the key precursor here. Let me make some space. I hope you understand all these things. And the key precursor here is viloxanthinine. Viloxanthinine. Sorry. So this is the key precursor, viloxanthinine. And if you find this is converted into xanthoxane, and this xanthoxane is finally converted into ABA. This xanthoxane is converted into ABA, and this conversion took place in cytosol. Okay, cytosol. So all these things will be very difficult to remember, but still do write it, because go through it. If you remember this, write these things in exam, you will fetch some extra marks. If you forget it, because there are a lot of things to remember, so just remember this MEP pathway. That would be enough. And the site of synthesis, obviously, this is green leaves, roots, and developing seeds. So this you should must write. Now, after occurrence and site of synthesis, the question I also asked us was transport. So you can mention things about transport. So it is a quite mobile hormone. It is mobile hormone, and it can be transported via both xylem and phloem. Okay. So through xylem, you can see when it is transported from roots to leaves, it is through xylem, and when from leaves to other parts, leaves to other parts, it is through phloem. Okay. And the transport is generally mobile, and it is also generally non-polar. And unlike oxygen, if you see oxygen is polar. Remember it, because they can give you the transport of oxygen also if you have given you the transport of ABA. So it is non-polar, and generally you will find them to be in transport. You will find them to be pH distributed or pH dependent distribution. pH dependent distribution, or you can say redistribution. pH dependent redistribution across membranes due to ABA's weak acidic nature, because ABA is weak acidic nature. So basically it can be pH dependent redistribution. Where there is high acidity, ABA can be distributed there to reduce the acidity. So these things can be mentioned. Now the fourth dimension in the bioassay of ABA. Bioassay of ABA. So first of all, you should understand the meaning of bioassay after uh, bioassay before writing about it, because many of us do not know about bioassay. Bio here will be a bioassay, so it gets difficult for us to understand or generate points. So bioassay is nothing but to detect or quantify, that is measure the quantity and detect the quantity of its concentration or its efficacy. All these things comes under bioassay. So how we can detect or quantify ABA will come in bioassay. The first bioassay, it can be done in kind of, you can mention stomatal closure bioassay. Stomatal closure bioassay, obviously, because ABA inhibits stomata opening. So you all know that ABA promotes stomatal closure. So what happens if stomata is closing? So you can search for ABA there, and if the stomata is closing due to stress condition, obviously there will be amount of ABA. So you can do this one. Secondly, cotyledon expansion test. Cotyledon expansion test can be done because ABA inhibits it reduces the expansion of cotyledons in various seedlings such as mustard, cucumber. In all these seedlings, the cotyledon expansion test can be done. We can also do uh, the bioassay of alpha amylase bioassay, because you will find that it inhibits the alpha amylase synthesis. Especially you will find them in barley, it inhibits them. So what happened if we it inhibits, and the degree of inhibition correlates to ABA concentration. If the inhibition is more, okay, if the inhibition is more, obviously ABA concentration is more. ABA concentration is more. If the inhibition is less, the concentration is less. So this can be done. We can also do seed germination bioassay, because also ABA inhibits seed germination also. So if the inhibition of seed germination is more, if the seeds are suppressed, the germination is seeds are suppressed very much, obviously the concentration of ABA is higher. So you can compare the ABA concentration in different extracts, seed extracts, stomatal extracts, cotyledon extracts, using these bioassays, and you will detect ABA, or you will quantify the concentrations of ABA. So this is how we can do bioassay. Now the fourth part of the question is physiological effects of ABA. Physiological effects of ABA. So first of all, we all know that ABA is known as stress hormone. So in the plants with stress, the amount of ABA will be high. Secondly, it leads to seed dormancy, enhances seed dormancy. Next, you will see that it leads to stomatal closure, just discussed. Enhances stomatal closure, especially under drought conditions, or when we have to reduce the transpiration and water loss. So there AVA can be of great use. Secondly, it also is a growth inhibitor. Growth inhibitor. So it opposes the reactions or actions of auxins and gibberellins. It suppresses cell elongation, reduces cell elongation. It also suppresses root and shoot development or division. Root and shoot division. So all these things can be mentioned. You can also mention here about it also leads to leaf senescence and abscission. Leaf senescence and abscission. Okay, it also leads to embryo and seed development various times. It regulates the maturation of embryo and storage protein synthesis. So you can leads to it regulates maturation of embryo and seed development. So you can mention this. Obviously, we have just discussed that it is stress hormone, so it has a role in protection of plant from abiotic stresses such as drought, salinity, cold, heat. So it induces the expression of stress responsive genes. So all these things you can mention. I think 5-6 points will be enough for the this answer. So we have discussed a lot amount of points here. We have discussed about occurrence, we have discussed about transport, we have discussed about site of synthesis, we have discussed about bioassay, and at last physiological effects. And in conclusion, you can write that how ABA plays a central role in stress physiology, growth inhibition, regulation of seed dormancy, and it regulates, or you can say it interacts with other hormones such as gibberellins, auxins, ethylines to fine tune the plant responses under changing environmental conditions. So that is all for today. If you like it, please press the like button, please do share, and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis, and if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.